I'm going to tell you a couple stories today. Basically, two different. So my lab develops machine learning methods primarily for both genomics and proteomics data. So I'm going to talk first about some techniques we developed for proteomics analysis, and then switch over to the genomic side of things. I recognize that some of these techniques may be more or less familiar to some members of the audience, and particularly mass spec tends to be less fewer people that know the details of how mass spectrometry works. So I'll try to fill you in on any details. Sorry for the moving furniture. Um, as I go along. And there we go. Okay, but as a motivation for the first part of the talk, we're going to talk a little bit about immunopeptidomics. So immunopeptidomics is basically the term for studying the antigen peptides that are presented by MHC. These are targets for immune cell recognition, so obviously very interesting from a biomedical perspective. In the context of mass spectrometry analysis of proteins or peptides in this case, these ones are difficult to analyze. One reason is that typically when you do a mass spectrometry experiment, the first step is the proteins are digested into peptides using trypsin, which preferentially cleaves at particular amino acids. And that has the nice side effect that it makes the analysis of the data much easier. When you don't have tryptic termini, there are many more possible peptides that are created, and so it gets more challenging. But also in this context, it's often the case that we care about individual mutations that occur. And so standard approaches to analyzing mass spec data that are based on using a reference proteome don't work very well for this kind of data. And so that means that for these kinds of data, often what people need to do is what we call de novo sequencing. So in de novo sequencing, the basic idea is someone's going to give you a mass spectrum. So in this case, the spectrum's on the left, and the x-axis there is the mass to charge ratio of individual fragments, ideally from a single peptide species that's been fragmented in the mass spectrometer. And the y-axis are the intensities of those fragments. And the idea is, given that sort of spectrum, or you can think of it like a fingerprint for that peptide, you need to infer what was the sequence that was responsible for generating the spectrum. So immunopeptidomics is one application of de novo sequencing. Others include things like metaproteomics. So in metaproteomics, if you're looking at soil or ocean water or what have you, you don't have a reference proteome. You don't know a priori what peptides to expect. And so you have to do de novo sequencing. Antibody sequencing is another, where the antibodies are created through the whole VDJ recombination procedure. And so you don't have a reference proteome there either. Other applications include just general peptidomics. Anytime you're studying peptides that are directly created by the cell, then they're not going to have these tryptic termini. They're not going to be as predictable. And then things like proteogenomics, where you're combining genomics with proteomics, but particularly often people are interested in detecting variants, and sequence variants are going to require that you do de novo sequencing. Paleoproteomics, looking at proteins extracted from ancient samples. Venomics, these kinds of things are all places where de novo sequencing is important. So my lab had worked in mass spectrometry for a long time, since 2003 or so, but it wasn't until around 2022 that one of the postdocs in the lab, Will Fondry, said, hey, there's this architecture that's been gaining a lot of popularity in the context of large language models called a transformer architecture. And he had the hypothesis that if we tried to use this kind of architecture for de novo sequencing, that might be a very powerful combination. And so he was just actually finishing up in the lab and leaving, but he continued to collaborate with us and with Meli Yilmaz, who was a PhD student at the time, to develop some software. And we collaborate with Wout and some undergrads in the lab, and Suwung Oh, who's an expert in machine learning and computer science. And what we produced is a piece of software that we refer to as Casanovo. And the basic idea is to treat this de novo sequencing problem kind of similar to a natural language processing task, right? So these large language models can translate between languages, by taking a sequence of words in one language and predicting the corresponding sequence of words in another language. And our idea is that we could do the same thing for translating from a sequence of peaks in a spectrum to a sequence of amino acids in the peptide. The difficulty being that in the case of mass spectra, the peaks correspond to either prefixes or suffixes, right? Every time you cleave a peptide, you get a prefix and a suffix. Well, so the prefix ones and the suffix ones are sort of overlaid on top of each other, and you don't know which one is which. So it's like you have two sentences, but they're written in opposite directions and then superimposed on top of each other. And then the usual problems. There's noise, there's variability, those kind of things that we have to handle. So I'm not going to spend a ton of time telling you the details of how this model works, except to try to give you an intuition about transformers. Casanova includes two different transformers in it. And the core idea of a transformer is that it learns sort of a matrix of what are called attention weights that keep track of important dependencies between observations in the data. And so the first transformer takes as input the spectrum, which is represented as a sequence of tuples. Each tuple just has a mass to charge ratio and an intensity. And the idea is that by looking at many millions of these spectra, the attention mechanism can pick up on important dependencies. Like, for example, the prefix and the suffix add up to the same total mass, right? And those pairwise relationships should be picked up. Or every time you have a particular amino acid, there's a difference in MZ that's characteristic of that presence of that amino acid. So the transformer encoder learns all that information, stores it in its weights, but it also makes what's called a latent representation, a vector representation of the spectrum that's passed to the second transformer, and that's the decoder. And this works in what's called an autoregressive fashion, meaning it's predicting in this case, one amino acid at a time. So in this particular case, you imagine that the model has already predicted most of this peptide sequence, PEPTID, and that's part of the input. And then it's being asked to predict what's the next amino acid. And it got it right. It got the letter E as the prediction. So it just autoregressively predicts one amino acid at a time. So we developed this model, we trained it on standard benchmark data sets and compared it to some state-of-the-art methods. In this plot, you're looking at precision on the vertical axis and coverage on the x-axis. So coverage, you basically imagine you get a bunch of spectra, you get a prediction for each one, but also a confidence, and you sort from your most confident prediction to your least confident. Clearly, the ones at the top of the list, you're more likely to get correct, right? So that's what we're seeing here in the top left corner here. You have very high accuracy. This is the vertical axis. What proportion of the things that you've predicted so far in the ranked list were correct? And then as you get further down the list, you get lower and lower peptide precision. So for comparison here, we have this, whatever that color is, greenish yellow line there, that corresponds to an existing tool called Novor, which uses a sort of more traditional machine learning model, sort of a state-of-the-art before the deep learning models came onto the field. And then there's DeepNovo, which was the very first method published in 2017 for using deep learning for doing the de novo sequencing task. And the difference between the red line and the blue line, that's DeepNovo and Casanova, is essentially the benefit that's brought by switching to this transformer architecture. So DeepNovo, for example, had just discretized the mass to charge ratio and made a vector representation from that. And if you know anything about mass spec, you know that the measurements are extremely precise. And so that discretization, we hypothesize, leads to a loss of information that we don't have to do when we do the transformer. The difference between the two Casanova lines has to do with the kind of data that they were trained on. The solid red line here versus the blue line, the solid red line was trained on a much larger data set and also higher quality data. And so that's sort of the take-home message from many machine learning projects, which is more data and better quality data leads to better performing tools. Now, we wanted to see what impact this had in practice. So going back to that immunopeptidomics example, we looked at some published data and we compared using Casanova versus trying to run a standard database search procedure on the same data. So this is only focusing on the immunopeptidomics results that don't have variants in them because they're in the reference proteome. But in that case, the solid line here, the gray line here, is what you get from doing the number of peptides that you can detect from doing a database search against this data using a standard 1% false discovery rate threshold. And if we say we run Casanova on that same data and just accept things that are in that set of detected peptides, that's what the dotted green line is. But if you then say, well, there's probably a lot of correct detections that are lower down in the list, below 1% FDR, and you accept those, that's the solid green line. And actually, to my mind, what's most important is the solid red line, which is anything that matches to the human reference proteome. Because if you think about it, given the search space for de novo sequencing of all possible peptide sequences, a random match to the human reference proteome is actually very unlikely from a statistical perspective. So you can show that it's very likely that the vast majority of the detections in that red line are likely to be correct. And of course, it's a big boost relative to the database search approach. Now, we also wanted to know whether those nominally correct detections have additional evidence that they're likely to be correct. And to do that, we turned to an existing machine learning model with the name NetMHC Pan-4.1. This is a model that was developed by a different group, and the basic idea of the model is to predict whether a peptide looks like it's likely to be an MHC binder, right? So in the vertical line here is the 500 nanomolar is the threshold for the prediction for when you call something as being, yes, it's likely to be a binder or not. And what you just see in this plot is a cumulative density of the binding affinity estimated by this machine learning model for 3 sets of peptides. Those peptides that were detected only by the database search, only by Casanova, or by both. And the nice feature is that you maybe can't tell, but there's 2 lines on top of each other, the red and the green, meaning that the ones that are predicted only by the de novo sequencing method have the same profile of predicted binding affinities as the ones that are agreed upon by both, whereas the ones that are predicted only by database search look much less like MHC binders. Now, after we published our first sort of machine learning conference paper on this, and unbeknownst to us, someone else grabbed our software and published a paper that we were gratified to see. This is basically someone took our software and a bunch of other pieces of software and compared them in their ability to do a different task, which is the antibody sequencing task. And so you can see these are precision recall curves. So recall on the horizontal axis and again, like precision or accuracy on the vertical axis. Comparing 6 different methods, the different plots correspond to different ways to do the digestion of the proteins into peptides. And the take home is that the solid line up at the top in each of these plots is the Casanova. So it really suggests that independent validation, that it's really giving you a big benefit. So shortly after this paper, the second paper about this software was published, one of the students in my lab, Justin, came to me and said, hey, don't you think it would be worthwhile to generalize this to work with data-independent acquisition data? So DIA data is kind of a big thing in proteomics mass spectrometry these days. The number of papers using DIA to generate their data has been increasing. And the basic idea of data-independent acquisition is that in a normal mass spec experiment, which is people call DDA, data-dependent acquisition, you first analyze a set of intact peptides, and then you isolate particular peaks that correspond to some peptide species of interest, and you fragment that species in a sort of directed fashion, and then you get a signature that corresponds to that one peptide species. That was like the sample spectrum I talked you through earlier. In DIA, you don't do that. Instead, you just fragment a large mass to charge range. Multiple peptide species get fragmented together, but you do it successively with the same predictable mass range for the precursors over and over again. And the result is that the signal associated with one peptide is, sure, it's mixed together with others, but it's also observed over multiple successive observations. So you get more complex spectra, but you also get a time domain that you can exploit that's not available in the normal setting. So we did develop a piece of software. It's called Cascadia that does work for this. And the main concept was to generalize the notion of a spectrum. So we're again going to do a transformer architecture. It's very similar architecture to Casanovo. And the main difference is how do we represent the input? So in this picture, you have what are called MS1 data. Those are spectra from intact peptides. And MS2 data, those are from fragmented peptides. And the red stripes correspond to the signal we would expect to see from one kind of peptide. So these red stripes here are repeated observations of what we call the fragmentation ladder. That's the pattern of prefixes and suffixes created by splitting this peptide into pieces. And the dashed lines up here are the isotope distribution, the three different isotopes eluting over time of the intact peptide. So we extract all of these data into one pseudo spectrum, which we call an augmented spectrum, and feed that into Cascadia. So whereas before you just had a MZ and an intensity, now you have the MZ and the intensity, but also this time index of where you are within the window, as well as a Boolean saying, are you an MS1 or are you an MS2? It turned out Justin had a really hard time getting this to train. If you just start feeding this to the deep learning model, it doesn't really, especially the Boolean features, confuse it. The time domain is hard for it to figure out. And so it actually took some doing, but Justin came up with a procedure for starting from a Casanova model with sort of simulating DIA-like dimensions for the DDA data to try to get it initialized and then gradually injecting real DIA data into the model until eventually it was able to learn these more complex four-dimensional input tokens. And once he got it working, it worked really well. So what you're seeing here is, again, precision on the vertical axis. But now, since we don't have a one spectrum per peptide kind of relationship, the x-axis is just the total number of peptides that were predicted. And again, we're using this intuition that if it matches to the human reference proteome and it's longer than length 7 or so, it's super unlikely to happen by chance. So we count those as nominally correct predictions. And what you can see is if you just take the DIA data and run it through Casanova, it really doesn't work well at all, which is to be expected because these data are way more complicated. But the lighter green line here is a variant of that, which a priori you might expect to work better. So there is a standard way in DIA processing for extracting what are called pseudospectra, where you basically look at the isotope distribution and do a deconvolution step to pull out all of the fragments that are associated with that isotope distribution. And that's actually how existing methods work. Some of the database search tools rely on these pseudospectra. And in fact, the DeepNovo DIA, which was the only existing deep learning method for analyzing this kind of data, also used pseudospectra as its input. So if we run Casanova on pseudospectra, it does give you a benefit, but not very much. Nothing compared to DeepNovo DIA, which was specifically trained for handling the pseudospectra from DIA data. And so these two curves here are two variants of that DeepNovo DIA trained on different data sets. But the main difference is that the problem with doing the deconvolution of making these pseudospectra is that sometimes you don't even see an isotope distribution, and yet there really is a peptide there. And in those cases, you'll never even make a pseudospectrum, and DeepNovo DIA has no chance to ever detect that peptide. And so that's why you can see that the Cascadia curve is way higher, is because there are a bunch of things that just got lost in the pre-processing, and that by just using this generic augmented spectrum approach, Cascadia was able to do much better. So again, we wanted to make sure that the things that we were saying were correct, we had additional evidence that they were correct. And again, we used a deep learning model for validation, but we used a machine learning model for validation, but we used a different approach this time. So here we're using a model called AutoRT. And what it does is it says, give me a peptide sequence in a charge state, and I'll tell you when is that peptide likely to elute off the liquid chromatography column. So one thing I haven't mentioned yet is that the first step in most proteomics experiments is you separate the peptides in a glass LC column first. And the order in which they come out is predictable based on the peptide sequence. So the idea is Cascadia has no knowledge of when the peptide came off the retention time column. So if its predicted sequence gives you a prediction that matches the actual time it was observed, then it's likely that it's a correct prediction. And so that's what we're seeing here is the observed retention times versus the predicted ones, supporting the idea that these detections are likely to be correct. Okay, so finally, the third piece of this story, of the proteomics half of the story, was again motivated by Justin. And he was asking whether or not we could repurpose the things that had been learned by Casanovo to solve other problems. And so this was a problem, a task that we did in collaboration with Wout Bitchermieux, who's a former lab member and now a professor in Antwerp, still with Suwang, but also with Nick Riley, who had some glycoproteomics data that he wanted help analyzing. So the basic setup here is that if you have a spectrum, there are actually multiple kinds of questions that you might want to ask about it. So the one we've been talking about so far is what peptide was responsible for generating this spectrum. But if you go in the proteomics literature, you'll see people writing papers about things like, is this a high quality spectrum? So is it even going to be identifiable? Or is this spectrum, does it contain a post-translational modification? Is the spectrum chimeric, meaning was it generated by more than one peptide or just a single peptide? Or is this from a cross-linked peptide? So a lot of people use peptide cross-linking to understand protein complex structures. And so in other fields of machine learning, there's been this notion of a foundation model, which is mentioned in the title of my talk. And I'm sure many of you have heard of it, but if you haven't, the basic concept was first proposed in the context, again, of natural language processing, where the idea is large language models like ChatGPT that I'm sure you've interacted with are trained in an unsupervised, or what is often called a self-supervised way, on tons and tons of data, nominally all the data that you can get from the internet, right? And they're trained in a very generic way, typically to predict what's the next word in the sentence. And those are surprisingly rich, they lead to surprisingly rich representations that capture important semantic relationships between words, and they capture sentence structure and all the sorts of things that you find when you can interact with a large language model. And to make it a foundation model, the idea is that it can generalize very easily to different tasks. So you can use it for writing poetry, answering multiple choice questions, doing document summarization. And often this can be done with very little training data because there's so much prior knowledge incorporated in that pre-training stage. And so the hypothesis in our case, in Justin's case, was maybe we could do the same thing for the kinds of tasks that people do apply to mass spectra. So the motivating example is going to come from Nick Riley's lab, where they study a particular kind of post-translational modification known as glycosylation. Imagine that you have a particular spectrum. It was generated using something called HCD, which is high collision dissociation. You do some real-time search and you can ask the question, is it glycosylated? Does it have a glycosylation somewhere in the peptide? But really what Nick's lab cares about is what kind of glycosylation does it have? There's two main kinds, N-glyco and O-glyco. And the reason it matters is because if it's an N-glycosylation, then the spectrum that you have in hand is sufficient to try to figure out what kind of glycosylation it is. But if it's O, then you have to spend the time to go and do an additional kind of scan called an ETHCD spectrum. So that's an important decision to be made in real time. So there are baseline methods that look at particular kinds of ions in the spectrum called oxonium ions. So you can take, for example, the ratio of two oxonium ions, and that's going to give you a baseline predictor. Here's a precision recall curve for that particular question of is it N versus O? And you can see it does a reasonably good job. We worked with Nick and trained a couple of machine learning classifiers. The first one uses XGBoost, which is a sort of off-the-shelf, very standard machine learning classifier, but trained on many different kinds of oxonium ions, and you can see it gives an improvement. But if you train one of these more sophisticated neural networks, like a transformer architecture, similar to Casanovo, it doesn't really improve relative to just the standard, more traditional machine learning model. And so the question was, could we do better? And maybe if we trained a foundation model, even though we have a relatively small amount of training data, the foundation model will allow us to do better. So that's the basic concept here. We're going to train a foundation model, and the hope is that in the pre-training stage, in our case, training Casanovo, it's going to learn stuff about mass and charge, fragmentation, isotope distributions, those kind of things. And then in the fine-tuning stage, where you train for the specific task you're interested in, it's going to learn about oxonium ions, the effect of glycans on fragmentation, and so on. So that's the main hypothesis. And the idea is you take the Casanova model and you just X out the second transformer. We're not going to use that transformer at all, but we're going to freeze the encoder. And then we're going to put a task-specific prediction head on. This is going to be a small predictor with a relatively small number of parameters that you can train quickly, even on a CPU. So there's a lot of compute that goes into this one, but this part is relatively cheap. And as a baseline, we're going to train an XGBoost classifier just on the task at hand, using just a standard spectrum representation with a discretized MZ axis. And then we're also going to train the full model, the transformer architecture, but trained without the pre-training stage. So I'll go through this quickly, but basically we revisited some of those tasks. For instance, we looked at the spectrum quality. So this is a published benchmark data set for predicting spectrum quality. You basically use database search as a gold standard and say, if you can successfully assign a peptide with high confidence to a spectrum, you say it's high quality, otherwise it's not. And here we're comparing the green line is a foundation model we've done the pre-training stage and then just trained the prediction head with the foundation model compared to training the whole model from scratch or training XGBoost. And you see there's a boost in performance there. Similarly, the chimericity task is this task of saying, is this peptide generated from one or more than one spectra? And you see a boost. But for some tasks, you're not really data limited. So we also turned to a published data set from a Nature Machine Intelligence paper on phosphorylation prediction, where they had 20 million training examples. So in this scenario, you've got enough training data that you don't actually expect the foundation model to be beneficial. But we use this as a way to demonstrate that if you randomly downsample your training set to smaller and smaller, we might see an increasing difference between the performance of the different models. So that's what we see here. This is a baseline model, just the XGBoost model trained from a standard representation of the spectrum. And the x-axis is the training set size. The y-axis is the area under the ROC curve. It's getting better as you get more data. If you use a more sophisticated model like a transformer, it's better able to make use of the really large data set. And so it works even better for large data sets. And then if you use your pre-trained representation, the key point is that over here on the left side, when you're lacking much data for this particular task, then you actually benefit from the pre-training a lot. Up here, it doesn't make much difference as there's sufficient data. So going back to the glycosylation task for Nick Riley's lab, the idea is that now we've got a green line here that boosts the performance on this particular task by a fair bit just by using this pre-training stage as our prior to training the model. So that's the main takeaway from this part of the talk, which is that you can take this pre-trained spectrum representation and repurpose it for a variety of different downstream tasks, and that in general, the foundation modeling improves performance on cases where you are data limited. Okay, so I'm going to switch over to the genomics stuff, but just want to say, if you do want to try out either of these tools, they're open source software. We have issue trackers, so if you try to use it and it doesn't work, please submit an issue and we'll try to respond and help you get it working. Okay, so now for the genomics side, what I'm going to talk about here is genome 3D architecture. So this may be more familiar to many of you, but just in case there are some people who aren't, the 3D chromatin architecture in the cell is hypothesized to be very important functionally. What we're looking at here is the inferred 3D structure of DNA in the nucleus of Plasmodium, the parasite responsible for causing malaria. And the hypothesis is that, and there's strong evidence to support this, that the genome 3D architecture is important for processes like gene expression, DNA replication, and so on. And so it's very interesting to study. One of the main techniques that people use to study genome 3D architecture is called Hi-C. It was first described in this science paper from 2009. And the basic idea is that you take cells and cross-link them, typically using formaldehyde, And then you introduce cuts, sometimes with restriction enzymes. There's other variants of the assay that use other kinds of cutting. Fill those cut ends in with biotin and then ligate them back together. Once you remove the crosslinks, you can then pull down and shear, then you can pull down the ones that contain the two different fragments that have been linked together. And that tells you that there was two different DNA loci that were in close contact in the cell. So the result of this assay is typically summarized in a matrix. Looks like this. It's a contact matrix. This particular one is from a yeast. And you're looking at the full genome on one axis and the full genome on the other axis. The strong diagonal here corresponds to the interactions within chromosomes, and the ones that are farther from the diagonal are interactions between chromosomes. But the main concept is that each count in this matrix is a count of how many times did you observe a paired end sequencing read that corresponded to this particular pair of loci. So for most of the contact matrices we'll talk about today, the resolution of this matrix is like 10 kilobases per bin. So you're asking how often did some read fall into this 10 kb region and the other end fell into this 10 kb region, and they were linked together. So one of the things that the field figured out sort of over time is that you can use this assay to capture phenomena at multiple scales. So, you know, in the initial paper, it was already immediately obvious that you could see this diagonally dominant structure in the genome-wide contact map corresponding to chromosome territories. Chromosomes like to interact with themselves, and they're less likely to interact with other chromosomes. But you could also do things like a eigenvalue decomposition and divide the chromosome into compartments, typically considered to be two compartments, sort of an active and a repressive compartment. Later, when the maps got more, when sequencing got cheaper and the maps got higher sequencing depth around 2012, people published papers saying, hey, there's actually, when you zoom in on the diagonal in the cis contact map, so within chromosome contact maps, you can actually see block diagonal structure there corresponding to structural domains, often called topologically associating domains. And then at the finest scale, you can also see loops, which often show up as punctate dots, which correspond to two loci that are sort of interacting from far away and forming a chromatin loop. Okay, so one of the funny things about this field is that you can ask different questions but about a high-C contact matrix, but they're often answered by totally different tools. So you can ask, where are the domains, or where are the loops, or where are the compartments? And you might have a different piece of software to answer each one of those questions, even though they're asking questions about the same data, just at different scales. So if you're looking at how reproducible two experiments are, you might look at using a random walk to characterize the similarity between two matrices. Or there's neural networks that have been developed for doing resolution enhancement. There are bespoke algorithms that were developed specifically for capturing these particular kinds of loop patterns that people see. And so our hypothesis, in fact, the hypothesis of one of the postdocs in my lab, was that we could try to use this this foundation modeling paradigm to create a model that he decided to call High-C Foundation that would exploit the same idea we talked about before. Namely, if you could just train one model that sort of learns what the structure of these contact matrices looks like, then it should be easy to fine-tune it to do a variety of different downstream tasks. Okay, so that the postdoc who came up with this idea is Xiao Wang, and it's a collaboration with Sheng Wang, who's an assistant professor in the computer science department. And then, of course, other members of my lab, and then also Sergei Dulatov, who needed our help to analyze some particularly challenging High-C data that his lab was generating. Okay, so similar to the previous part of the talk, there's a sort of with any foundation model, there's often a two-stage approach to the training. So the model here, High-C Foundation, is trained in two stages, and the basic unit of analysis in all of these, all of this work is going to be a submatrix, where we pull out a matrix that's actually 2.2 megabases by 2.2 megabases, where the matrix is at 10 kb resolution. And in the first stage, we're going to just learn from the Hi-C data itself. So we downloaded all of the data from all the data we could find from the ENCODE consortium, from the 40 nucleome consortium, but also from other published data sets. We do this pre-training stage, and then we're going to separately do fine-tuning for a variety of downstream tasks. Okay, so how do we do the pre-training? Here, rather than natural language processing, we're borrowing techniques from image analysis. And the basic idea is to use a model architecture called a vision transformer. So it still has this notion of a transformer in it. So you're still learning pairwise attention relationships. But instead of saying we're going to predict the next letter in a sentence or that kind of thing, we're actually doing masking. And so the input submatrix is one where we've intentionally hidden some of the data. So here's an example of a masked submatrix. You take this submatrix, you concatenate it all together into one big long vector, and then drop out all the masked regions, keeping track of where they were, of course, and then put that through an encoder. What that does is it makes a latent representation, which you are going to, that's the thing you want to be very useful, right, for later steps. And then there's a decoder that then tries to reconstruct it. And the learning objective is basically to make sure that the reconstruction is accurate, that you accurately capture the things that were masked out. Now, an important feature of this model, you'll notice the encoder is drawn as if it's much bigger than the decoder. And that's because it actually is. We really care that this representation is quite rich. And so we don't want a lot of the work to be done by the decoder. We make it a lightweight decoder, and that forces the encoder to take on most of the modeling work. And that's important because later we're going to toss out that decoder. We don't actually care about being able to reconstruct masked submatrices. What we care about is being able to represent the information in the Hi-C matrix in a concise format. So let's look at some of the tasks that we looked at. So the first task is reproducibility analysis. So here the question is, I've got two replicate experiments. Did I get a good enough replication of my experiment that I can treat them as if they're a single experiment? Or maybe more importantly, I've got a bunch of related cell types. I've done Hi-C on each, and I want to know what's the relationship among those. Which of these experiments are more similar to the other? There's actually a literature, there's 6 or 7 different methods that have been proposed for computing similarities between Hi-C matrices. In our case, we used a contrastive learning approach. So we trained a prediction head. You're always using the frozen encoder, but now the representations from that encoder are being fed into basically this little decoder that's trained with this particular kind of contrastive loss. And the idea is you put in triplets, 2 biological replicates and 1 non-replicate. And you want it to be the case that in the embedded space that this model is producing, the replicate ones are close together and the non-replicates are far apart. And if to the extent that they're not close enough for the replicates or not far apart enough for the non-replicates, you push or pull them apart, put them together, pull them apart using the optimization strategy. The standard way that this is usually analyzed in the literature is like this. So what you're seeing on the x-axis is the score from our particular similarity measure, the Hi-C Foundation Learned Similarity Measure. And the vertical axis here are different biosources. So you can think of them, in this case, different cell lines. And the 2 kinds of dots are whether this pair of experiments is a biological replicate or a non-replicate. Okay, and obviously what you want is for there to be a separation between the 2. What you can see is in each case, all the replicates are much more similar to one another than the non-replicates are. And in practice, what you often report is the so-called BR-NR margin. How well did we separate the 2 in a scale when all the different measures are between 0 and 1? So, and one important point that's worth mentioning is that when we compare to state-of-the-art methods, we're actually always looking at novel biosources, so cell types that weren't used during pre-training, but we're also looking at chromosomes that were not used in pre-training. So we split the chromosomes and only trained on a subset of them. For all these tasks, we're looking at new biosources and new chromosomes. Here, we're comparing to 3 of the existing state-of-the-art methods in terms of this BR-NR margin, And we also always compare to what's called a no pre-train baseline, where you just train the whole model from scratch on this particular task. And what you can see is even when we're looking at novel biosources and novel chromosomes, we do a much better job of separating than the state-of-the-art methods. The next task we looked at was loop detection. So this is one of the more challenging tasks because you typically require quite high coverage in order to see these loops. It's why they weren't really described in great detail until about 6 or 7 years after the first Hi-C paper. Because in the older Hi-C matrices without much coverage, they really aren't very visible. And so the idea here was that we were going to take biological replicates and we're going to train our predictor to be consistent across different mixtures of two biological replicates. So you make a sort of pseudo gold standard by combining the two replicates and then running an existing loop caller. We used one called hiccups to call loops. And then you're basically downsampling, giving it much less data, but different mixtures of the two replicates and trying to train the model to always make those same predictions again. So it's a way to make the model robust to lower coverage and also to be consistent for variance of the same input data. So we do that for the fine tuning, and then at inference time, we just basically make predictions and then run a little what's called a mean shift cluster, just a clustering algorithm to actually make the predictions of where the loops are. And what we compare against is the same model hiccups, but also two others, mustache and chromocyte. And here the score that we're using is the so-called loop F1 score. So it's basically a combination of the precision and the recall. And what you can see is we're comparing both to the state-of-the-art methods and the no pre-trained baseline. And in each case, it's doing a much better job of identifying loops. The third task that we looked at is resolution enhancement. This is actually one of the problems that we had tackled ourselves. So Sheng Wang and I started collaborating by writing a paper just about doing the resolution enhancement problem. But Hi-C Foundation improved upon even our own method. And the basic idea is that in our case, we were again going to use this downsampling idea to train the model. That is, we're going to say we want to be able to enhance the resolution of a given Hi-C matrix. It's kind of like, those spy movies where you get a picture of the license plate, but you can't quite read it, and you click, enhance, and then suddenly you can see them. We want to be able to see the loops that weren't visible before, or the boundaries of the TADs or the domains and so on. And so we train the model by basically pretending that we don't have as much coverage. We cut it down by a factor of 4 or 5, and then we try to predict what the real data would look like, and do that over and over again. I'm not going to go through all the different performance measures. This is a super well-studied problem, so there's a lot of existing methods out there, and there's actually a lot of different performance measures as well. Some of them capture things like reconstruction error, so then you can look at like the mean squared error, for example. Some of them capture the correlation, and so then we have Pearson correlation and Spearman correlation. And some of them look at how well you can do downstream tasks, like can you do a better job of finding loops, or can you do a better job of finding boundaries between domains? In all of these cases, these performance measures, we're comparing to two different variants of the Hi-Carn method and then a method called Hi-CNN and Hi-CSR. Across all of these different performance measures, we're outperforming them. This over here is the rank mean, which is just where you take the rank for all the different measures and average them together. So it sort of summarizes all of the results in one. Okay, so then we wanted to turn to tasks that hadn't been tackled previously in the literature. And so we tried something new that hadn't been done before. And honestly, I sort of told Xiao, it's kind of a dumb thing to try. Why are you bothering? But actually, it worked way better than I expected. Better, well enough that it was worth reporting in the paper, which was, he said, let's just see if we can predict some of these other epigenomic signals. So CTCF, or histone modifications like H3Q4ME3, or even local chromatin accessibility as measured by a TAC-seq or DNA-seq. And the idea was, we're going to take a submatrix of input Hi-C as embedded by Hi-C Foundation and see if we can train a predictor to output these kinds of sequences. I was surprised when he showed me this. Again, this is on, data that it's a cell line that was never seen during training. It's on a chromosome that was never seen during training. And you're getting here a Pearson correlation of 0.55 for the DNA-seq measurements. The challenge here is that there's nobody else has tried to do this before, so we don't have any state-of-the-art methods to compare against. The only thing we could compare against was sort of a simple baseline that we came up with, which was based on doing sort of a PCA and then trying to reconstruct from that. Or, of course, training the whole thing from scratch without any pre-training stage. And so, not surprisingly, we can do better than those relatively simple methods. But to me, it was quite exciting that you could even get this information out of the Hi-C matrix. And then finally, the last thing that Xiao wanted to do was looking at single cell Hi-C. So you can do Hi-C analysis in single cells, but the challenge in the Hi-C space is that you don't get that many reads, right? If you look at state-of-the-art Hi-C matrices today for bulk Hi-C, they'll have a billion reads typically. Some of them have multiple billions of reads in them. Whereas single cell Hi-C matrices have on the order of 100,000. If you're really lucky, hundreds of thousands, right? But it's a much, much sparser type of data. So it seemed unlikely that we were going to be able to make much sense of it, particularly because when you analyze single cell Hi-C data, you don't do it at 10 kb resolution. It just doesn't make sense. There's not enough resolution. So people will analyze these matrices at like 500 kb or megabase resolution. So what Xiao did, which is a little counterintuitive, was he said, well, sure, I've got these 2.24 megabase by 2.24 megabase submatrices, but they're at 10 kb resolution. If I switch to 1 megabase resolution, that submatrix now becomes an entire chromosome. So I'm just going to train from single cell data. I'm going to do my fine tuning, that is, on single cell data that is measured at 1 megabase resolution, even though my pre-training was at 10 kb resolution. So that's what he did. And he did the single cell enhancement task. So it's similar to the task we talked about before, where you simulate downsampling the data and then re-predict the full data set. And you can evaluate how well that works by comparing again to some existing methods. So these are 4 different single cell Hi-C data sets. Of course, none of them were used in pre-training because it's single cell data. But again, we restricted ourselves only to the test chromosomes. And we're comparing to existing methods, SC Hi-C cluster, Higashi, and SCVI 3D. And in each case, whether you're measuring by Pearson correlation or there's actually 6 other performance measures in the supplement, but they're summarized all in the rank mean in the second one, you see substantially improved prediction performance. You can also do this by looking at the pseudobulk level. So now you can say, I'm going to enhance my single cell Hi-C data and then ask how well does it capture what I would have seen if I did a bulk Hi-C experiment. So for this evaluation, you have to have single cell data and bulk data from the same kind of sample. And what you can see visually at the top is that the two agree quite well. And also at the bottom, you can see that we're performing better than the no pre-train and the raw baseline as we vary the number of counts per cell, so the sequencing depth of our data, and also the number of cells that go in. So in this case, we're generating pseudobulk profiles from only like a couple of hundred cells, and you're still able to get a pretty good estimate of what the pseudobulk profile looks like. So the basic idea here, what our goal is that Hi-C Foundation can be kind of like a Swiss Army knife for any kind of Hi-C analysis where the input is a contact matrix and you want to ask any kind of question about it. I would argue that unless you have a ton of data, you're probably going to be better using the representation of your data that's been passed through our encoder to solve whatever problem is you want to solve than if you try to do that directly from your data without this encoder. So in fact, the paper has some additional analyses. So for instance, the neutrophil data that Sergei Dulatov's lab had analyzed, they really needed help because they couldn't get very many cells. So they had basically low input Hi-C data. And we were able to help them basically find loop structure that was not visible in their data just initially. And we also have analysis across high-C data generated from many, many species, hundreds of mammalian species, showing that this is useful in that context as well. So, and again, the software is publicly available, so I encourage people to try it out. None of this would be possible without all the people in my lab who do all the work. So this is a picture of the current lab members. About half of these people work in genomics and multiomics analysis, and the other half do the proteomics stuff. In addition, of course, I'm grateful to the NSF and the NIH for funding and to you for listening, and I'm happy to take questions.